Welcome. Let us discuss about ultrastructure of muscle and chemical basis of muscle contraction. Comparative physiology. Cardiac muscle. Found only in the heart. Cardiac cells arranged in branching pattern. In each cardiac cell, one or two nuclei, striated, involuntary, speed of contraction not as fast as skeletal but faster than smooth muscle. Smooth muscle found in the walls of the hollow organs like stomach, intestine, blood vessels, ureter, etc. Smooth cells are fusiform in shape. In each smooth cell, one nucleus is present, non-striated, involuntary, speed of contraction, slow and wave-like. Skeletal muscle attaches to a skeleton. Skeletal cells long and cylindrical. In each skeletal cell has many nuclei. Striated, voluntary, speed of contraction capable of rapid contractions. Ultrastructure of skeletal muscle. Epimysium. The entire muscle is surrounded by an external connective tissue wrapping. Fascicles. Bundles of individual muscle cells in skeletal muscle. Perimysium. Each fascicle is surrounded by a connective tissue layer. Endomysium. Within the fascicle, the third connective tissue layer wrapping around each individual muscle cell. All the three connective tissue layers binds the muscle cells together, providing strength and support to the entire muscle. The muscle is attached to the bones by the tendons. It is a fibrous cord of inelastic fibrous connective tissue. Myofibril. Thread-like fibrils that make up the muscle fiber or muscle cell or myocyte. Myofibril contains myofilaments. Myofibril have sarcomeres. It is a distinct anatomical and contractile unit of thick and thin filaments. Myofilaments. Myosin, the thick filament, and actin, the thin filament. They are responsible for muscle movements. Muscle fiber is enclosed by a plasma membrane called sarcolemma. Cytoplasm of muscle fiber is called sarcoplasm. Within sarcoplasm, T-tubules, transverse tubules, allows the transport of substances through the muscle fiber. Sarcoplasmic reticulum, membranous structure surrounding each myofibril, stores calcium, helps transmit action potential to myofibril, forms chambers, terminal cisternae attached to T-tubules. Triad, formed by one T-tubule and two terminal cisternae. Muscle striations. A striated pattern within the myofibrils. Thick filament. Myosin, A band, dark band. Thin filament. Actin, I band, light band. M line. At the center of A band or at the middle of the sarcomere. Z line. At the center of the I band or at the two ends of sarcomere. Zone of overlap. The densest and darkest area where thick and thin filaments overlap. H band. Area around M line has thick filaments. Titin. Connects myosin to Z lines in sarcomere. It is very elastic, able to stretch 3 times its elastic length. Calcium ions released by sarcoplasmic reticulum causes thick and thin filaments to interact. Muscle contraction is caused by the interaction of thick and thin filaments. Actin filament. Helical structure contains myosin binding sites made up of G-actin molecules. Two strands of G-actin molecules are twisted together with two regulatory proteins, namely tropomyosin and troponin. Tropomyosin, rod-shaped protein. It occupies the groove between the twisted strand of actin molecules. It blocks the myosin binding sites on the G-actin molecules. Troponin, a complex of three globular proteins. One is attached to actin molecule, other one is attached to tropomyosin, and the third one contains binding site for calcium. Myosin filament contains tail, rod-like, central portion, binds to other myosin molecules. Head, face outwards in opposite direction, made up of two globular protein subunits. One, binding site for actin. It interacts with actin during contraction. 2. Binding site for ATP. The enzyme ATPase is located for the hydrolysis of ATP. Mechanism of muscle contraction. Muscle contraction is initiated by the signal from central nervous system, CNS. Release of acetylcholine at the neuromuscular junction. Causes action potential in muscle fiber. Triggers the release of calcium ions from sarcoplasmic reticulum. Calcium ions combine with troponin and tropomyosin uncovers the binding sites on actin and initiates the contraction. Myosin binding sites on actin is exposed. Myosin head binds to actin forming cross bridge. After cross bridge formation, the ATP which is present in the myosin is used to cock the opposite direction from its resting state. As the ATP is used and the ADP plus P is released and power stroke occurs as the myosin pivots towards the M line. When another ATP molecule attaches to the myosin head, the cross bridge between the active site of the active molecule and myosin head is broken, thus freeing up the head to make another bridge and complete the contraction. When the signal from CNS stops, calcium ions are pumped back into the sarcoplasmic reticulum. Tropomyosin masks the binding sites. Filaments are pulled back to the original position. Relaxation.